Welcome to Leafing Through Science, where we dive into the pages of popular books in the life sciences with the writers who made them happen, from new releases to timeless classics. I'm your host, Hedi Young, and this is part 2 of my conversation with Gareth Leng, author of The Heart of the Brain, the Hypothalamus, and its Hormones. If you haven't yet listened to part 1, I heartily recommend you do, but here's a quick refresher. In the first episode, Gareth took us deep into the remarkable world of neuroendocrinology, the study of how hormones and the brain interact. We zoomed in on a tiny but mighty part of the brain called the hypothalamus, a behind-the-scenes operator instrumental at every stage of life, from birth and puberty to sex and parenting. We also met the different clans of neurons in the hypothalamus that produce unique cocktails of hormones and peptides, like the so-called love hormone, oxytocin, and learned how these signals send commands to the rest of the body via the pituitary gland. And we looked at a crucial feature of hormone signaling, the rhythmic pulses that drive behaviors such as breastfeeding. In this second part, you'll hear how these clans of cells can change the very language they speak, orchestrate your internal clock, and even tip the scales of your body weight. So, from circadian rhythms to the science of obesity, settle in as Gareth leads us further on our journey into the hypothalamus. Now, while oxytocin tends to get all the attention for its role in bonding and reproduction, vasopressin, which we've already mentioned, plays a crucial role in something much more down-to-earth but absolutely essential: regulating how much water our bodies hold onto. It's released when we're becoming dehydrated, and it acts on the kidneys to reduce water loss by concentrating the urine, essentially helping us hold onto water when we need it most. So, Gareth, could you briefly describe how vasopressin helps the body balance hydration and how this beautifully regulated system works?
How vasopressin acts at the kidney was really unveiled by the revolutionary discovery of another molecule called aquaporin, which is a molecular shuttle that vasopressin activates this molecule in the kidney cells that shuttles water across the cell membranes. It's this basic mechanism that allows water to be retrieved. We take these things for granted. So many things we take for granted. We think, I can drink a glass of water, and what consequences does it have, except I feel a little bit better? But our bodies have to maintain a stable volume and a stable blood volume. And they have to maintain a pretty stable electrolyte balance. And so the salt we take in and the water we take in has to be dealt with. If you exercise, you will lose salt and you will lose water. If you drink too much water, you've just got an expanded water volume. You can get rid of it in urine, but whenever you get urine, you lose salt a bit. If you can't find water, you have to retain water massively. But if you retain water and you carry on eating, then your salt content will go, and you can't get rid of the salt without producing urine. So your body is constantly confronted with these dilemmas. It's not so simple. So vasopressin has got a really important role when there is a sustained dehydration, when water is in low availability, or when the only water that's available is salty. In both those conditions, and they're quite different challenges, you've actually got to get rid of the salt but retain the water, and that's vasopressin that is important for doing that. Now, you're going to need that for over a prolonged period. If you're deprived of water or can't access it for a long time, it's a problem. So you need to maintain vasopressin there for a very long time, potentially. So the pituitary has this huge store of vasopressin in the pituitary. It's got enough, actually. If you stop synthesizing, it's got enough to last for nearly a month. Sounds a lot, really. But actually, if you need to increase your vasopressin a lot to get that water retention, if you need to increase it tenfold, then that big store will only last you a few days. You really have to look after it, and you have to be very efficient how you use it. And the curious thing about the vasopressin cells is each one of them will fire in bursts of spikes. The bursts last about 20, 30 seconds maybe, and then they're silent for the next 30 seconds. Then they burst again. In this way, they're kind of bursting cells, like the GnRH cells. But there is a difference, because the GnRH cells were all synchronized. They're synchronized by the kisspeptin signal, and they were all bursting together. The vasopressin cells don't burst together. So they all burst at different times. And so there isn't a pulsatile release of vasopressin out into the blood. There's just a background release. And that was intriguing. And so that made people think, why are they firing in bursts? Because in the end, vasopressin is just coming out as a stream. You need it all the time. You need it at a steady level. You don't want it in pulses because actually, oddly, the kidney receptors for vasopressin, they don't desensitize. They're very unusual. So why are these cells firing in pulses? Well, it turns out that actually there's another advantage of firing in bursts, and that is that actually you can secrete those vasopressin vesicles more efficiently if you fire in bursts of spikes than if you fire continually in a background level.
Sorry, this is what you call the optimization principle, the idea that
That's right.
brain cells releasing vasopressin don't just fire off signals on their own, but work together to share the burden of activity, taking turns like runners in a relay to avoid burnout.
Well, that is indeed the next level of it. So individually they fire in bursts because bursting is very efficient. But they also take it in turns to, as one neuron will get exhausted, will release its burden, then it will fall quiet, and others that have had the chance to recover can take over. So again, it's the heterogeneity of the population that helps sustain an efficient pattern of secretion over a prolonged period of time.
Yeah, individual vasopressin neurons, as you say, they may fire inconsistently or not at all. But when you look at them as a group, a different picture emerges, and it's the collective activity that seems to carry the real signal.
That's absolutely right. And again, it comes back to the metaphor of the clan that I introduced, which is really that to understand the vasopressin neural system, you can't really consider a vasopressin neuron on its own. You have to understand how the whole population is acting together. It is as though this is the unit of vasopressin. It is the population, not the single cell.
And so far, we've talked a lot about how neurons in the hypothalamus send signals using pulses, peptides, and coordination across networks. But what's even more remarkable is that some of these neurons can actually change what they're saying and who they're saying it to, depending on what's happening elsewhere in the body. In response to signals from the body's periphery, like hormones coming from the adrenal glands or the gonads, certain hypothalamic neurons don't just change their firing patterns, they switch the very chemical messengers they produce. For example, during stress, some neurons stop releasing CRH, that's corticotropin releasing hormone, which kicks off the body's stress response, and start releasing vasopressin instead.
Well, yes, I mean, the story that you've alluded to was such a shock at the time. It was just early in my career when that story was unfolding, and it was absolutely extraordinary. I'm talking about the time when CRH was first discovered, and this is the releasing factor that controls the release of a hormone from the pituitary, a hormone called adrenocorticotrophic hormone, or ACTH, and this is the hormone that controls the activity of the adrenal gland. The adrenal gland is a gland that produces the glucocorticoid hormones that we release in response to stress and in response to inflammation and other things. So it's an extremely important system, very important for our health, indeed for our survival. And so Wiley Vale, back in the late 70s, early 80s, identified CRH as being the releasing factor for ACTH. But up until that time, there was a large group of scientists that thought there was good evidence that actually the releasing factor for ACTH was vasopressin. Not the vasopressin that we've been talking about that's released from the pituitary, but vasopressin that's produced by a different population of hypothalamic cells that is released into these blood vessels that supply the anterior pituitary. So, were they completely wrong? Well, then they showed something really interesting, which is that the two factors act synergistically together. So if both are present, then the effect on the pituitary is bigger than the sum of their separate actions. They have a combined, very powerful effect if they're both present at the same time. Well, that was kind of interesting. And the next stage really was, well, what happens if you adrenalectomize a rat? Now, adrenalectomizing a rat cuts out the negative feedback.
Adrenalectomize, removing the adrenal gland.
Adrenalectomy is removing the adrenal gland. When glucocorticoids are released, then there's a negative feedback on the brain to reduce the production of CRH. So if we take away that negative feedback, what happens to these populations of cells in the hypothalamus? And it was assumed that if you take away this negative feedback, you'll get a big CRH signal. That's not what happened. The conspicuous thing that happened really is that you get a big vasopressin signal. And the cells that were making CRH now seem to be making vasopressin. So they switched. And that was kind of extraordinary. So suddenly, this new population that we'd found could change to a different population, could change from being a CRH population to being a vasopressin population. And that normally we always have these two populations working in different balance side by side, but under different physiological conditions of stress, continuous stress, chronic stress, or whatever. The proportions of these factors that are made will change. Well, that was a shock, of course. We had assumed that given a brain at birth or whatever, it doesn't change very much. And the idea that actually the chemical content of those neurons changes and the signals they use changes was profoundly disturbing, I think. But at least we thought then that, well, it's another kind of curious oddity of the hypothalamus and no general significance. Well, of course, yeah. Now we know that actually all of these messengers, all the peptides that produce in neurons, they're very, very variable. They change under physiological circumstances. The amounts that neurons make change. So the chemical phenotype of cells is a very plastic thing.
Yeah, amazing flexibility. It's almost like these clans can switch languages.
That is right. Some people have even thought it's possible that oxytocin neurons can change into vasopressin neurons, and vasopressin neurons can change into oxytocin neurons. Though most vasopressin cells make just vasopressin, maybe a tiny little bit of oxytocin, and most oxytocin cells make just oxytocin, but maybe a tiny bit of vasopressin. There are a few cells that make about equal amounts of each. But if you stimulate one of the system, then that changes, and you get more of these cells that make both. And so even these two populations that seem to be doing quite different things, there seems to be a possible element of interchangeability between them.
Now, let's talk about something that affects every one of us every single day, our internal body clock, or what scientists call circadian rhythms. These are the roughly 24-hour cycles that control things like sleep, body temperature, and hormone release. At the heart of this system is a tiny part of the hypothalamus called the suprachiasmatic nucleus, or SCN. In the 1980s, scientists made a remarkable discovery. If you damaged the SCN in rats or hamsters, their circadian rhythms would disappear completely. But if you then transplanted a new SCN from a newborn animal into the brain, the rhythms came back, as if the brain had a new internal clock installed. This was a huge moment. It confirmed that the SCN really is the brain's master clock, and it also sparked excitement about the idea that tiny pieces of brain tissue, like the SCN, could be transplanted and still function in the host brain. So, Gareth, can you tell us more about what makes the SCN such a powerful timekeeper and what these experiments have taught us about how the brain keeps time?
One of the things that's particularly interesting about those experiments is that different animals and different strains of the same animal have slightly different timing to their circadian clock. Now, the way we know we've got an intrinsic circadian clock is if you keep a normal animal in constant light or constant darkness, then actually their body rhythms will still follow an approximately 24-hour rhythm. Right? But usually it's a bit longer than that. Usually it's more like a 26-hour rhythm. And what happens is that the light cue every morning re-entrains that rhythm. It pulls it into line, so that keeps it to the 24-hour clock. But the intrinsic rhythm is longer than that, and different strains of animals have got slightly different timing. So you've got some that have got a much longer intrinsic rhythm, more like 28 hours or 30 hours, and some which is very, very close to 24 hours, or even a bit shorter. So if your strain has got a fingerprint, if you like, for its intrinsic rhythm, what happens now if you damage the suprachiasmatic nucleus, as you described, and you now transplant the suprachiasmatic nucleus from another animal? Will it show the same rhythm that the recipient animal had before the lesion, or will it show the timing of the transplanted animal? Well, that showed very clearly that the rhythm is a property of the transplanted tissue. It's not a property of the physiology of the recipient, it's a property of the tissue itself. So that timing is encoded within that scrap of tissue that regenerates. So that's the first real clue. So you have to then find the understanding of that clock mechanism by understanding that isolated tissue. And we now know what's understood to be what's called molecular clockwork. There are a series of genes that produce things that interact with delays that result in a rhythmic production of particular genes by neurons in the suprachiasmatic nucleus. And so we now have what we think is a kind of molecular explanation of this intrinsic rhythm. And it's a lovely, beautiful story. But there's another excitement about those original experiments, which has opened up the real possibility in people's minds that you could get restoration of function after a brain injury by some transplant procedure. In order to get this, it was assumed that these neurons that you transplant have got to be integrated appropriately with the neurons of the host brain. They've got to make the right connections and be sensitive to the right signals to function in this way. And people started to try and look for that, but actually they didn't really seem to find much evidence of interaction of the transplanted tissue with the host tissue. It was a puzzle. And then Ray Silver over in the States performed an absolutely beautiful and extraordinary experiment, really. When she transplanted the suprachiasmatic nucleus, she enclosed the entire transplant in a semi-permeable membrane, enclosed it in a capsule so that no fibers could get in or out of that tissue, but that liquids and things in solution could diffuse between them. So it could be oxygenated and still get supply of metabolites from the blood, but it couldn't make any connections. And the extraordinary thing was that even this transplanted nucleus that's enclosed in its plant worked. The circadian rhythms were restored. And that was extraordinary because that meant that actually this key signal that goes to the rest of the brain that drives our rhythms doesn't travel by conventional neural pathways at all. So we now know it travels by diffusible hormonal signals coming from that explant. It was almost such an extraordinary experiment, extraordinary outcome, that really it was scarcely believed after that for another 20 years. It was known about, but nobody wanted to talk about it because it was just such a challenge to our understanding.
Yeah, amazing how you can insert a brain from another animal that's enclosed in its own membrane, and yet it still has this effect. Beautiful, beautiful experiment.
Yeah, absolutely beautiful experiment.
Taking an experiment out of your book, Gareth, because another fascinating piece in this story is how our internal clock, the SCN, responds differently to light depending on the time of day. You describe a striking finding from your research in rats. When light is given at the end of the night, it triggers the release of vasopressin in the SCN. But if the same light is given at the start of the night, it doesn't. And that might help explain why, if we see light early in the morning, we might wake up. But if we see it earlier on in the night, we often stay asleep.
That story really came from a discovery of a colleague of mine, Mike Ludwig, that there are cells in the retina that produce vasopressin, and that these cells project specifically to the suprachiasmatic nucleus. And so they don't project the conventional light processing parts of the brain, but they do project just to this area that's involved in the circadian rhythms. And this is a vasopressin projection, so they're producing vasopressin. So again, I think this came from the idea that the problem with peptides as signals is that they can't be replaced rapidly. They've got to be synthesized and it takes time to be traveled down. They're big vesicles, they're expensive to produce. Neurotransmitters can be recycled very rapidly at synapses, so you can have a rapid turnover. But peptides are a big and precious message. So if you've got a signal that's being secreted throughout, say, the light period for 12 hours in a day, you're going to get progressive depletion of the store of vasopressin in that pathway. And in the dark period, you'd expect it to recover and come back. And that is almost kind of inevitable, really, if you've got a continued stimulus for secretion. So what are the implications of that? The implications of that seem to map onto exactly what you're saying. That a light signal which is going to be releasing vasopressin when the stores of vasopressin are high, will have a different effect at the end of the light period when the stores of vasopressin might be low. So that seemed to kind of match up with that observation.
And goes to show how storage of these molecules is really important to their function. Because, as you say, unlike fast-acting neurotransmitters like glutamate, which are always ready to go and quickly recycled, vasopressin is stored in these larger, slow-to-replenish vesicles, and these build up over the night. By the end of the dark phase, they're fully stocked and ready to be released. But earlier in the night, there's simply not enough available to trigger a strong response. You briefly mentioned the pineal gland in the book, which the 17th century French philosopher Descartes famously believed was the seat of the soul and the point where mind and body meet. My own father actually spent 4 years in the 1980s researching the pineal gland and whether or not it played a role in triggering puberty. But because the method of measurement they were using, the gas chromatography mass spectrometry assay, wasn't working properly, they didn't have reliable data to go on. Turns out that unlocking the secrets of puberty is a tricky business, especially when your machine refuses to cooperate. So, Gareth, let's turn now to the topic of obesity, which you explore in depth in the book. It's often framed as a simple matter of willpower. Eat less, move more. But the reality is a bit more complex. And you write about the importance of genes and the environment we're born into, and even the bacteria in our gut. You mentioned one study where researchers transferred gut microbes from obese and lean human twins into germ-free mice. And amazingly, the mice that received microbes from the obese twins gained significantly more fat, while those given microbes from the lean twins stayed slim. And one theme in the book is that appetite and sexual behavior are closely linked. They actually influence each other. So, Gareth, how does the hypothalamus balance these two drives, and why might hunger or nutrition affect things like libido?
So when we started to work on the melanocortins, they were produced in the hypothalamus, and these are again peptides. One of the peptides, called alpha MSH, was already kind of known as being a very potent appetite inhibitor. And so if you're looking for a therapeutical treatment for obesity, an appetite inhibitor is probably an interesting thing to look at. And so we started to look at this. We started to look at its cellular actions and where it was expressed and how it was regulated. There's a big pharmaceutical company, Merck, and Merck, of course, had a big program of their own on the agonists for this receptor. But their first, if you like, preclinical trial, fairly simple. They gave it to a group of volunteer students, I think. They gave this drug to them, and they were obviously looking for effects on appetite. But what they discovered was that these young men actually went quite red in the face and started crossing their legs because they had erections. So this came now as being a problem, really, and it now turned out these drugs that they were working on, these MC4 agonists, or these melanocortin agonists, had three potential actions. If they worked on some melanocortin receptors, they'd produce a tan. If they worked on others, they would reduce appetite. And if they worked on others, they would increase sexual libido, especially males, probably females as well. It sounds like a dream.
So
Of course, for a pharmaceutical company, this is not a dream at all. This is a horrible mess.
It's incredible. Different effects could be giving you a tan or increasing your libido.
Yeah. But the MC4 ones particularly have effects both on appetite and libido. And ones with a different receptor profile will affect your periphery and give you a tan. Yeah. But it's this, but really they were keen on kind of separating out these two things. Either of these might be interesting as a therapeutic target, because of course there are conditions of low libido and infertility because of that. And of course there are problems of weight balance. But you don't really want to mix these two up. And so really kind of interested in knowing what the mechanisms were and could they be separated. The obvious suggestion for us is that maybe these were actually acting on the oxytocin neurons, because we already knew then the brain oxytocin system, those brain oxytocin neurons, were involved in erection and involved in libido and involved in sexual response in females as well as males. So we already knew that oxytocin was very tied up with sexual behaviours. But there was also evidence around then that it might actually have a role in appetite regulation as well, in suppressing feeding. So could both of these things be explained by an action on oxytocin neurons? And that was the line that we pursued. To quite a large extent, that's probably true. I think it's now accepted that a lot of the effects of the MC4 agonists on sexual behavior are apparently mediated through the oxytocin system. And at least some of the effects on appetite are likely to be as well. So these are entwined. Now, I said this is a bit of a mess. It's a bit of a problem for a pharmaceutical company because they like to be able to cleanly separate them. And I think at that time, we kind of looked at this and said, maybe you're not ever going to be able to separate these. Because if you look back in evolutionary terms, even in very simple organisms, then there is a very, very tight link between appetite for food and appetite for sex. When you go back into very simple organisms like the annelid worm, C. elegans, and C. elegans got very simple behaviors. Essentially, if it's hungry, then its priority is food, and it will go and graze on available food in its environment. And when it's sated, then it will display what's thought to be an analog of sexual behavior, display leaving behavior. It'll leave that patch and go off in hunt and search for a mate. So that's its priority. If it's short on food, it needs food, and that's its priority. Once it's got food, now its priority is sex. And so it's the same brain areas in C. elegans and in other species that control these, and they control them in a reciprocal fashion. When you switch on the motivation for one, you switch off the motivation for other. I don't know if you've noticed, but it generally is true that eating food and having sex are things that you do at different times. You know?
Yeah,
you don't
want to confuse your appetites.
You definitely don't confuse your appetites. That's right. And it's also the case that if you make an animal hungry, even slightly hungry, by delaying its mealtime, then it will be slow to engage in sexual activity. And we also showed through studies in rats that if you give hungry rats that signal from alpha MSH into the brain, then that will stimulate their sexual activity even though they're hungry. So we think that there is, and it's probably evolutionarily conserved across a wide range of species, there is this close relationship between these key motivations. I mean, these are key motivations. They're fundamental to our survival, for food and for reproduction. And obviously, you've got to survive. So if you need food, that's got to come first. Once you've got food, now the priority is to reproduce.
And finally, Gareth, you traced the story of the discovery of leptin, a hormone produced by fat that tells the brain how much energy is stored. But as you explained, the key isn't just having leptin, it's whether the brain can respond to it. In leptin resistance, the brain acts as if we're starving, even when there's plenty of fat.
I think we're all kind of familiar with diabetes, type 2 diabetes, and we're all familiar with understanding and recognizing that it's a disease. Type 2 diabetes is primarily a problem of resistance to insulin. So its overproduction of insulin results in desensitization of the receptor mechanisms, and eventually the body becomes insensitive to insulin. No question it's a disease. It's a long-lasting, very, very serious disease. In many ways, obesity is a disease in the same way as type 2 diabetes is. Because we've now recognized that one of the key signals that regulate our appetite is this hormone leptin. Now, leptin is produced by fat cells. If we put on more fat, we produce more leptin. That feeds back to the brain to say, look, we've got enough fat, you don't need to eat anymore. If we eat anyway, if we ignore that signal, then what will happen is that we'll progressively become desensitized to that leptin signal. It's exactly analogous to what happens in type 2 diabetes. And the consequence now of that insensitivity to leptin is that that signal doesn't exist. That normal physiological restraint on our appetite and eating becomes weakened and becomes absent. And that, as you know, now becomes very, very difficult to reverse. So if you call diabetes a disease, really, you have to call obesity a disease in the same way. It's got a physiological substrate. And if we understand that mechanism, now we can understand at least how genes in part affect that disease thing. Because all the pathways through which leptin signal are now subject to genetic change and variability from individual to individual. So, and now we know, in fact, that the gene defects anywhere along that pathway from leptin to the brain systems that regulate feeding. Mutations in any of those pathways can result in a disordered body weight. Now, people in the obesity field really have just stopped talking about it being a problem of willpower. They've been quite frank. The reality we've known for a long time, of course, is that the biggest predictor of how your own body weight, your body weight or my body weight, is the body weight of our parents. 80% of our body weight is heritable. We've known that for a long time. That doesn't mean to say that if we massively overeat, then we can't shift that body weight, become very, very overweight as a result of excessive eating. All I'm saying is that it's just not so simple. And for most people, they actually maintain a very, very stable body weight, even without thinking about it. My body weight has changed by a tiny amount over 50 years. Your life changes in so many ways. Your activity levels change. You have children, you have stress, you have breakdowns, you have workloads, you live in different environments, you have passions, you drink too much or whatever. And through all of that, we keep generally a phenomenally stable body weight. We drift very, very slightly up. I think over those years, I've put on something like a few hundred grams a year over 40 years on average. And you can work out what is that in terms of excess food. And I worked out in my case, it's something of the order of half a banana a week. That is what, if you like, the excess food intake that accounts for my weight gain. And yet that small weight gain over 40 years puts me in the overweight category, from well in the normal weight category, now in the overweight category, not in the obese category. But really, the big picture is not how overeating affects our body weight. That's really rather a boring picture. The really big and exciting picture is how on earth do we keep our body weight so stable for so long in a very, very changeable environment and different challenges in our life? And how do people differ in the way that they can maintain a stable body weight?
And again, like the water regulation case and vasopressin that we discussed earlier, it's another example of a fundamental feature of life that we often take completely for granted. At the heart of these functions, stability and regulation of the body is the hypothalamus.
Indeed.
So, Gareth, what was your favorite part of the book? Did researching the book change your opinions on any of your work or surprise you at all?
I think that's the reason for writing a book, really. Writing that book for me is like having a long conversation with yourself. It's indulging that, really. You can only have a long, extended argument with yourself. Nobody else has got the patience to be there and go through that. And that's what you have to do when you write a book like this. You have to be satisfied that you've taken the argument to a point that you feel you can show it to other people. And so, yes, for every page, you read 50 new papers and you're constantly discovering new things and you're testing things out. And that is always an interesting and exciting point. And as you go, of course, you change. If you're not interested in the possibility of being wrong and excited when you are wrong, you shouldn't be a scientist. And so actually, as you work and your ideas change and you discover that, what thought probably is wrong, that's what science is about, and it's got its own excitement. So, of course, you go back, and if you were to write it again, it would be different. I think I had a lot of fun writing it, enjoyed writing it. It was a lot of work. Learn about a lot of things you didn't know about. It made me think hard about evolution. And I think probably that, for me, was the most challenging and interesting thing, to engage with the idea of how these systems could have evolved. I think if you're going to understand the brain, if you're serious about it, you have to understand how it functions, how the elements work and how they interact and how they do what they do. But that's not enough. You've also got to understand how it's developed, how a small number of genes somehow build this thing that works so robustly. And that's really important because you're not going to understand so much of how it functions is determined by the fact that it's self-organized from a small set of genes and a small set of instructions. It's a big constraint on the kind of brains we have. And the other element, of course, is how it evolved. And we know that now the peptide systems are really kind of ancient. You can find basically oxytocin neurons in primitive mollusks. The history goes way back before. Not so long ago, oxytocin was the hormone of love, or the hormone of lactation. It seemed like a quintessentially mammalian hormone. But now you can trace its origins back to mollusks.
So
All
how many
through
millions of years are we going back to the
400 million. Well, it's been traced back to the common ancestor of all animals with a common body plan.
and
So
we're
we can, of course,
talking, more than 400 million
yes,
years ago.
yeah, that's right. Oxytocin cells, as we know them, that control the pituitary, are basically the same in all mammals, but they're basically the same in all vertebrates. So you've got to look before the vertebrates to find where they came from. We know that before the vertebrates, there was a single ancestral peptide that was duplicated and gave rise to oxytocin and vasopressin. So that was a difference that happened around the time of the emergence of bony fish. But we're now talking about reaching long, long before then to the origins of these systems.
A story as old as time itself. So from one book to another, then, we always ask guests to recommend a book, be it fiction or non-fiction, to our listeners. Do you have a book that you'd like to recommend, Gareth?
To be honest, I mean, there are several books that really made a big impact on me. And they're not new books, they're old books. There are two, really. One's a philosopher, Karl Popper's book on the logic of scientific discovery. And the second was Kuhn's book on the structure of scientific revolutions. And I read those as a student of mathematics. And it was those books that made me want to be a scientist. And I've gone back to them again and again for the contrasting but exciting pictures they painted of science. There's another book that I discovered just recently. I wrote this with one of my sons, Roger Leng, and this came out a couple of years ago, The Matter of Facts. Writing this book, which is a book about the nature of science from the perspective of a scientist, which I am, and Roger's is as a social scientist. It is that discussion between us, looking at things from two different perspectives, and it's a product of that. And in the course of that, I discovered a book that I should have read and never did until then. And it's by a French scientist called Bruno Latour, who is very well known in France. In fact, he's very well known throughout the world of social science. He's, I think, the most highly cited scientist, full stop, in the world. But his really breakthrough into recognition was a book called Laboratory Life. Now, that is really very relevant, which is why I should have read it, because Laboratory Life is the account of his time in the laboratory of Roger Guillemin. And Roger Guillemin is one of the people that won the Nobel Prize for identifying GnRH that we've talked about. So he spent a year in that lab in the Salk Institute, really as he described it, as an anthropologist. He was like a fly on the wall, just watching how scientists went about their work. And he produced this book called Laboratory Life, which I read and is absolutely wonderful. I found it transforming. I found it funny. You could certainly understand why it made him famous, but it gave such a fresh insight into what science is really like that it was stunning. So, Laboratory Life by Bruno Latour.
Well, my reading list just got quite a bit longer. So the hypothalamus is sometimes disparagingly being called the lizard brain, which I think is unfair to both lizards and brains. So hopefully today we've given the hypothalamus the respect that it's due. Gareth, thank you so much for this wonderful conversation. And to everyone listening, thanks for joining us and see you next time.
Thank you very much.